In this video, we learn more about the process of gene translation. During translation, the information that is stored in the sequence of bases in the mature mRNA is used to synthesize protein molecules within the cytoplasm of the cell. The ribosome, which is a complex made up of both protein and RNA, catalyzes the translation of the mRNA sequence into protein. In eukaryotes, transcription and translation are separated because eukaryotes have a nucleus where transcription and RNA processing take place, and then the mature RNA transcript exits the nucleus into the cytoplasm where proteins are made. So how does an mRNA triplet specify an amino acid? To address this question, there were two hypotheses that were proposed. One stated that the mRNA codons and amino acids interact directly, whereas the second, that was proposed by Francis Crick, stated that an adapter molecule holds amino acids in place while interacting directly and specifically with a codon in mRNA. So there was an adapter molecule serving as a connection between amino acids and the codons in mRNA. This adapter molecule was later found to be a small RNA called transfer RNA or tRNA. As shown in this graphical representation of the two hypotheses, according to the first hypothesis, scientists proposed that amino acids somehow directly read the information in mRNA, codon by codon, and combined with each other to form a polypeptide. On the other hand, Francis Crick proposed that there were adapter molecules that were reading the information in mRNA codons and using that information to catalyze the formation of peptide bonds between amino acids, constructing a polypeptide chain. Subsequent experiments conducted with radioactive amino acids revealed that they are actually lost from those molecules called tRNAs and incorporated into polypeptides synthesized within ribosomes. This inspired the use of the term transfer to refer to these RNA molecules because amino acids are transferred from RNA to the growing end of a new polypeptide. These experiments also confirmed that amino acyl tRNAs, meaning tRNAs that carry an amino acid, act as interpreter molecules in the translation process. These were Crick's adapter molecules. When they conducted this experiment, scientists wanted to ask the question, what happens to the amino acids attached to tRNAs? They hypothesized that tRNAs carrying these amino acids transfer the amino acids to growing polypeptide chains. To address their question, scientists set up an experiment where they added tRNA molecules that were attached to radioactively labeled amino acid leucine to a reaction solution containing the ingredients that are required for translation to take place, including mRNA and ribosomes. They then observed what happens to these radioactively labeled leucine molecules. Their prediction was that the radioactive amino acids will be found in proteins. If this was not true, they would not be able to find any radioactivity in proteins. So they measured the amount of radioactivity in both tRNAs and proteins over time and found that initially, radioactivity was high in the tRNA molecules, and over time, that radioactivity decreased, while radioactivity within proteins increased. This indicated that the amino acids were transferred from tRNA molecules to protein molecules over time, because their evidence supported their hypothesis. They concluded that amino acyl tRNAs transfer amino acids to growing polypeptide chains. Over the next set of slides, we'll look at each of the ingredients that's needed for protein synthesis. We've already looked at mRNA molecules that are processed in the nucleus and are transported into the cytoplasm for protein synthesis. Other ingredients that are required for this process include charged tRNA molecules or amino-acyl tRNAs and the ribosome, which catalyzes the synthesis of peptide bonds between amino acids. The process of attaching an amino acid to tRNA requires the use of ATP. This reaction is catalyzed by enzymes called amino-acyl tRNA synthetases that charge the tRNA by catalyzing the addition of amino acids to tRNA molecules. There's a different amino-acyl tRNA synthetase for each of the 20 amino acids. There are also one or more tRNAs that associate with each of the 20 amino acids. A tRNA molecule covalently linked to its corresponding amino acid is called an amino-acyl tRNA. So if we look at this image of a tRNA molecule, we can notice some unique characteristics, including its shape, which is in the form of an inverted L. This is true for all tRNA molecules. The tRNA is single-stranded, and the strand folds upon itself to form this secondary structure, the base pairs within which hydrogen bond with each other. At one end, the sequence CCA binds amino acids when the tRNA molecule is charged, and at the base, there is a sequence that recognizes the codon within mRNA. This sequence on the tRNA is called the anticodon. This sequence allows the correct amino acid to be incorporated into the protein. In this image, you can see the enzyme amino-acyl tRNA synthetase that catalyzes the attachment of an amino acid to the tRNA molecule in order to charge it and form the amino-acyl tRNA that then participates in the process of translation. So how many tRNAs are there? There are a total of 64 codons, 3 of which represent stop codons that don't code for any amino acids. That leaves us with 61 different codons that code for amino acids. But there are only about 40 tRNAs in most cells. Based on findings such as a tRNA with an anticodon GUU being able to pair with both CAA and CAG codons in mRNA led Francis Crick to propose the wobble hypothesis, which states that the anticodon of tRNAs can still bind successfully to a codon even though the third position does not quite match the typical Watson-Crick base pairing. This allows one tRNA to base pair with more than one type of codon. In addition to amino-acyl tRNAs and mRNAs, an additional requirement for translation is that of ribosomes, which contain both protein and ribosomal RNA, which is another type of RNA. Ribosomes contain two subunits: the small subunit, which holds the mRNA in place during translation, and the large subunit, where the peptide bonds form. During translation, 3 distinct tRNAs line up within the ribosome. All 3 tRNAs are bound at their anticodons to corresponding codons within mRNA. The sites where these 3 tRNAs bind within the ribosome are called the A site, which is the acceptor site for amino-acyl tRNA, the P site, where a peptide bond forms that adds an amino acid to the growing polypeptide chain, and the E site, where tRNAs no longer bound to an amino acid exit the ribosome. This cartoon image shows the 3 distinct sites occupied by the 3 tRNA molecules: the A site, which accepts an amino-acyl tRNA, the P site, where a peptide bond is formed between the incoming amino acid, which is added to the growing polypeptide chain, and the E site, where tRNA molecules that have already given away their amino acids exit the ribosome. Panel B shows a model of the same ribosome during translation. Translation can be divided into 3 phases. namely initiation, elongation, and termination. Let's look at each of these phases separately. Translation initiates at the codon AUG, which is called the start codon. This codon codes for the amino acid methionine. In bacteria, the start codon is preceded by a ribosome binding site. This is a sequence that the ribosome first attaches to and then starts translating from the start codon. This sequence is called the Shine-Dalgarno sequence, named after the scientist that discovered it. It is complementary to a section of one rRNA in the small ribosomal subunit. Interaction between the small subunit and the mRNA is mediated by initiation factors that aid in the recognition and binding to mRNA. In bacteria, translation is a three-step process. The first step involves the binding of mRNA to the small ribosomal subunit. The initiator amino acyl tRNA bearing N-formylmethionine or FMET binds to the start codon, after which the large ribosomal subunit binds, completing the formation of the complex. Translation is now ready to begin. So to summarize what we saw on the previous slide, the small ribosomal subunit, with the help of initiation factors, binds the mRNA molecule at the ribosomal binding site, or the Shine-Dalgarno sequence, after which the initiator amino acyl tRNA carrying the amino acid methionine binds to the start codon, and then the large ribosomal subunit binds, completing the ribosomal complex. An incoming amino acyl tRNA can now occupy the A site. The second phase of translation, known as elongation, involves three steps. This phase begins with the arrival of a new amino acyl tRNA, followed by peptide bond formation, and then translocation. At the start of the elongation phase, the initiator tRNA is at the P site. The E and A sites are empty. When the next amino acyl tRNA occupies the A site, the anticodon within the tRNA base pairs with the codon within mRNA that specifies a particular amino acid, which in this case is glutamate. After this, the ribosome catalyzes the formation of a peptide bond between the initiator methionine and glutamate, followed by the translocation of the initiator tRNA, which is now uncharged, into the E site in order to exit the ribosome. The second amino acyl tRNA bound to glutamate now enters the P site, leaving the A site empty for the new incoming amino acyl tRNA that recognizes the third codon within mRNA. The third amino acyl tRNA, now carrying the amino acid arginine, recognizes and binds to the third codon within mRNA at the A site. A peptide bond is formed between the second and the third amino acid, followed by translocation, which triggers the exit of the uncharged initiator tRNA, movement of the now uncharged second tRNA to the exit site, and the translocation of the third amino acyl tRNA into the P site, allowing the entry of a fourth amino acyl tRNA into the A site. This process continues till a stop codon is reached. Since the active site of the ribosome is entirely composed of ribosomal RNA, which catalyzes peptide bond formation, the ribosome is considered a ribozyme, which is an RNA with catalytic activity. As translation continues, the ribosome moves along the mRNA, recognizing one codon at a time and recruiting new amino acyl tRNA molecules to add amino acids to the growing polypeptide chain. Translation stops or terminates when the A site encounters a stop codon. This causes a protein called a release factor to enter into the A site. Release factors resemble tRNAs in size and shape, but they don't carry an amino acid. These factors catalyze hydrolysis of the bond linking the tRNA in the P site with the polypeptide chain. This leads to the release of the polypeptide chain from the ribosome. As we can see in this image, when a stop codon reaches the A site, a protein called the release factor, which is shaped just like a tRNA, enters the A site. There still exists a bond between the tRNA occupying the P site and the polypeptide chain. This bond is hydrolyzed by the release factor, after which the now uncharged tRNA molecules are released from the ribosome. The polypeptide chain is also released from the ribosome, and the ribosome itself disassembles. The two subunits separate and the mRNA molecule is also released. Let's look at an animation of the process. Translation begins when a tRNA carrying the amino acid methionine binds the start codon within the mRNA. The large ribosomal subunit then binds, completing the ribosomal complex. Subsequent amino acyl tRNAs then bind the A site, translocate to the P site, where peptide bond formation takes place, and then exit through the E site on the ribosome. The anticodon of each tRNA recognizes and binds to the complementary codon on the mRNA. The amino acid carried by each incoming tRNA is then chemically linked to the growing polypeptide by the ribosome. This process continues until a stop codon is reached, at which point a release factor enters the A site, hydrolyzes the bond between the amino acyl tRNA and the peptide chain, and leads to the disassembly of the ribosomal complex. After translation is complete, most proteins go through an extensive series of processing steps called post-translational modification before they're ready to perform work within the cell. Proteins called molecular chaperones speed the folding of proteins. This folding determines the protein's shape and therefore its function. Many proteins are altered by enzymes that add or remove a phosphate group. Changes like these often switch the protein from an inactive state to an active state or vice versa. This concludes this section on translation. After going through this information, I'd like you to be able to respond to the questions listed on this slide. Thank you.